Cell transport material using two methods. First one is passive transport, and the second method is active transport. So let's talk about passive transport first. So passive transport requires no or little energy. So there's just a little bit of energy that is required by the cell for material transport, and molecules move from higher concentration to lower concentration. So we have cell membrane right here. So this is the structure of cell membrane. You learned about it in your last lesson. It is lipid bilayer, and material passes through cell membrane, and it is selectively permeable. That's another thing you learned in your last lesson. So let's say you have molecules on one side of the membrane, and then there are more molecules on the other side of the membrane as well. And let's say you have more molecules on the left side, and there are less molecules on the other side. So in passive transport, molecules always go from area of high concentration to area of low concentration. So in this case, molecules will travel in this direction. They will transfer till equilibrium point is reached. Equilibrium point is a point where there are equal number of molecules on both sides. And passive transport is done in three ways: diffusion, osmosis, and facilitated diffusion. So three methods. First is diffusion. The second method is osmosis. And the third way is facilitated diffusion. So in diffusion, molecules move from area of high concentration to area of low concentration. So the key term that you need to remember is that molecules move. Osmosis is same thing as diffusion, but it's diffusion of water, not molecules. And then diffusion is, again, it's from area of high concentration to area of low concentration. So water moves into the cell and dilutes the concentration in the cell to more closely match the outside of the cell. So let's look at some examples of osmosis before we talk about facilitated diffusion. So this is an example of movement of water from area of higher concentration to area of low concentration. So first type of solution we have is hypertonic solution. So in this case, water is transported out from the cell because there is less water outside the cell. And solute concentration inside the cell is lower. So there is less solute particles which are present inside the cell. So therefore, to maintain equilibrium, water is transported out of the cell. Hypotonic solution is opposite of hypertonic. In this case, solute concentration inside the cell is higher. So there is a lot of solute that is present inside the cell. Now water will move in to dilute it until equilibrium point is reached. So remember, in hypertonic solution, you have less solute inside the cell. So there's a less solute inside the cell. Water is transported out to balance it out, to balance everything out. In hypotonic solution, solute concentration is higher inside the cell. So there's more solute. So to balance, to reach equilibrium, water goes in to dilute the solution. Isotonic is amount of water transported into the cell is equal to the amount of water transported out from the cell. Solute concentration inside the cell is equal to the solution outside the cell. So these are the three examples of osmosis. The third type of diffusion is facilitated diffusion. So in this case, we need to talk about proteins that are present in plasma membrane. So the structure that I have drawn before, I didn't draw proteins in it. So let me show you how proteins look like in plasma membrane. So this is cell membrane again. And then inside the cell membrane, there are proteins, and they're called protein channels. So these right here are proteins, and they're called transfer proteins. So proteins allow for transportation of highly specific molecules. So in this case, transportation is selective. Only certain molecules will pass through protein channel. This is passive transport. That means diffusion is going to be from higher concentration to lower concentration again, and very less energy is going to be required. So the only difference between the diffusion osmosis and facilitated diffusion is that in this case, protein channels allow the molecules to move, and channels are very selective, what molecules can pass through it and what molecules can't pass through it. So now let's talk about active transport. So active transport is the opposite of passive transport. In this case, energy is used by cell for material transport. And in this case, molecules move from area of low concentration to higher concentration. Since the molecules are going to move from lower concentration to higher concentration, which is against the concentration gradient. So a lot of energy is required for active transport. And sometimes molecules need to fuse to take it inside, and it is called endocytosis. So let's talk about endocytosis. Okay. So first of all, let's look at the term endocytosis. Endo means in. Okay. So you have your cell membrane again. And then the molecules that needed to be transferred, or that needs to be transported from one location to another, they are going to fuse with the membrane. So let's say this is a molecule that needs to be transported. This is a molecule right here. It needs to be transported. So it is going to fuse together. And then it's going to come out of the other side. It's going to cross the cell membrane, go to the other side, and it's going to come in the form of vesicles. And that's how the molecules are being delivered. So the molecules will actually fuse with the cell membrane, and then it goes to the other side, and it is delivered as vesicles. Um, so another type of so there are different types of endocytosis. So let's talk about the two types. So there is phagocytosis. In this case, amoeba is an example of an organism which uses phagocytosis. So amoeba pulls in what it needs to take and engulf. So let's look at the diagram right here. So this is the diagram of phagocytosis. So this is amoeba. And what happens is this molecule right here needs to go in. So it's just like, it looks like amoeba is opening its mouth. So what's really happening is the membrane surrounds amoeba. Sorry, the membrane surrounds the molecule, engulfs the molecule. It just fuses with the membrane, and then it is released into the organism. So phagocytosis is a cell engulfing large particles. Next, another example of exocytosis is pinocytosis. So what happened in pinocytosis is a cell engulfing small particles. They are most common in cells of multicellular animals such as humans. So these are the particles that needs to go in, and the cell is going to engulf the particle. So this is step one, and then the particle is engulfed by the cell membrane, and then it forms into vesicles. And the difference between phagocytosis and pinocytosis is phagocytosis is a cell engulfing large particles, and pinocytosis is cell engulfing small particles. So that's the difference between them. But their process is the same. The particle is going to fuse with the membrane, and then it will turn into vesicle, and then it's delivered as a vesicle. There's another process that cell uses. It's called exocytosis. Exocytosis is reverse of endocytosis. Exo means exit or leaving. And a cell uses this process to get rid of cell waste. And what happened in this case is, let's say you have a cell membrane right here. So this is the particle that cell needs to get rid of. It is going to fuse with the membrane, and then the particles just go out. And this is step 3.